Hello everyone, welcome back. The purpose of today's lecture and homework is to give an introduction to recombinant DNA technology with the purpose of moving into the next 2 weeks of class where we're going to do some cloning experiments. This particular page here, introductory page, shows you on the left the very first time that different pieces of DNA were fused or ligated together. And so that's a famous experiment. We'll come back to that and talk a little bit more about it in the lecture. And then on the right shows you a picture of humulin, which is human insulin that has been cloned. And because it's cloned, it can be expressed at high levels, which makes it useful. And it also can be modified so that different amino acids can be changed in the protein to make it better, like more soluble or whatever purpose the company needs. Okay, so let's talk about gene cloning. So here first, let's go through a couple of definitions. We're going to be talking about gene cloning, so copying a very specific part of DNA, something that would be an open reading frame for a protein or some other aspect that you're interested in. Comparing this to cloning of organisms, of course, which would be creating a genetic copy of an individual. So that's not what we're talking about here. We're talking about at the DNA level. The methods that were necessary for cloning were developed mostly during the 1970s. Of course, they've been greatly improved since then, and things have been made a lot more standardized and easier. But what we're at least going to start with was developed in the 70s and then started to get used and adapted in laboratories all across the world during the 80s and 90s. And now pretty much almost every cell and molecular biology lab does some aspect of cloning. Cloning, being able to copy a gene, is super useful for the purposes shown here. Basically, I don't have to read through this, but you can. It's useful, of course, for experimental purposes like sequencing the DNA or making mutations, generating probes. It's also useful so you can express the genes for use in experiments or even express proteins and bits of proteins for use in treating human health, which is a big thing, obviously. To clone a gene, it involves at least two different DNA sources. One of them is your target gene or target DNA, which here is called the experimental DNA. That can either be chromosomal DNA or cDNA, as we'll talk about in a second. And then there has to be some sort of vector DNA if you want to be able to copy the clone. The vector is a plasmid or something that allows you to put the DNA segment into either bacteria or eukaryotic cells or even viruses so that it can be copied that way. To get the DNA, the scientist has to do work in the lab, obviously, from obtaining the tissue of interest to extracting from the lysed cells either RNA, which would be used to make cDNA, or genomic DNA. The vector carries the piece of DNA of interest, but then the vector has to go into a cell. And the cell that holds the vector DNA is called the host. Almost all the vectors used now in cloning were originally derived from natural sources. So bacteria carry plasmids, and they can exchange those plasmids. And on the plasmids, they typically carry genes of in use to the bacteria. One of the biggest ones, of course, are antibiotic resistance genes or genes that express restriction enzymes. Other vectors that are in use developed initially from viral DNA that had promoters and different aspects that are useful for expressing genes in cells. So now most vectors have that were derived from these original sources have been modified by mixing and matching a bunch of different DNAs together to make something that's super useful. The picture shown here shows you a plasmid. The gray arrows in the middle labeled A, B, C, D, and E are genes of a variety of different uses. One of them is beta-lactamase, which is used for antibiotic resistance against ampicillin. Then there's the lac repressors being expressed here, so you can control gene expression and a variety of different things. Over on the right-hand side of the plasmid, let's see if you can, shown here, that area is called the multi-cloning site, and packed into that one small little area are multiple cloning sites, not one, EAG1, etc. And this makes it easy to clone your DNA of interest at the very C terminus of that other gene labeled C. And in this case, that gene is glutathione transferase. And so this is a plasmid that's used by a bunch of different labs to make what are called GST fusion proteins. So let's talk now about how you can start putting these two DNAs together, this particular plasmid shown here, and then any DNA that you're interested in. You have to be able to cut the DNA because it's either already circular, like a plasmid, or a chromosome, which is linear but really, really big, too big to clone. So you want to take your small piece and insert it. In most cases, you're going to be using a restriction enzyme to cut the DNA at a very specific place. Restriction enzymes, there's thousands of them, and they recognize very specific sequences, as I'm sure you guys are all aware. In this example here, we're cutting two different DNAs, one on the left in blue, one on the right in purple, with an enzyme called EcoR1. It recognizes the 6 nucleotides, G, A, T, T, and C. And then when it cuts, it generates what's called a 5' overhang. So you can see that easily on the left in the blue one after you've cut with the enzyme now. You have the nucleotides T, T, A, which are linear and hanging over the edge of the DNA. When you have this, you can mix two different pieces of DNA together that are complementary to each other, and they will stick together. So enzymes that cut and leave overhangs generate what are called sticky ends. And so those two DNAs will bind to each other, as shown in the third line. The blue and the purple are binding to each other, but they are still not completely joined. Notice there's a little gap between the G and the A on both strands. That has to be sealed together with phosphodiester bond, and that's done in this case by an enzyme called DNA ligase, just like in real DNA replication. So the DNA ligase would come in, generate the covalent bond, the phosphodiester bond between the G and the A, and now you have fused together two pieces of DNA. Restriction enzymes were discovered in the 60s and 70s initially by this particular group here, Arber, Smith, and Nathans. But now many thousands of DNAs have been, restriction enzymes have been found. They're produced naturally by many different bacteria, and the bacteria have evolved these genes to help fight off typically invasion by viruses, where the viruses that inject their DNA into the bacteria. The bacteria try to destroy that DNA by cutting them up with restriction enzymes. You can buy a lot of these restriction enzymes, as we'll see soon, commercially, and there's interesting software that's used to be able to analyze your DNA for specific restriction enzyme sites. Not so long ago, if you were working with restriction enzymes, you had to be able to purify them yourself from whatever bacteria you were working with. So that's sort of an old school skill. Here are some examples of restriction enzymes, some common ones, like on top, BAMH1. You may notice if you look at the names of the enzyme and the name of the bacteria that's where the uh, names come from. So BAMH1 is from Bacillus uh, amyloliquefaciens. Sorry, that's a big one. Anyway, so the BAMH1 enzyme is produced at least from that bacteria initially. It also recognizes a 6 nucleotide sequence, but in this case the sequence is GATCC, and it cuts between the two G's. And so this one would also generate a 5' overhang. Not all of them have that particular pattern, so if you look down on the list to NAE1, it's a blunt cutting enzyme, so it cuts between the C and the G in that particular sequence there, and generates a blunt in, piece of DNA rather than one with sticky ends. So just to review a little bit here, here's ECOR1, the one that we started with. It cuts this GAATTC between the G and the A, and when it cuts, it separates the DNA strands and generates a 5' overhang, as shown there. I mentioned the NAE1, it's the blunt cutter. Here it cuts between the C and the G, and as you notice on the bottom right, when it does that, it generates two pieces of DNA. Each of them, which are blunt, so there's no overhang. These can still be cloned. It's just a little more difficult than using a restriction enzyme that has an overhang. So let's use an example and go through the steps for gene cloning. And as our example, we're going to clone the beta globin gene into a bacterial vector, plasmid vector. And the vector that we're going to work with has two important genes. One's called R confers resistance to ampicillin, so it's going to be labeled as AMP-R, and then the other one expresses an enzyme called beta-galactosidase, which is the LAC-Z that you're probably already familiar with. Both of these genes can be used to help select the proper plasmids after we've done the cloning. One of the issues when you do the cloning is that you're going to put them back into bacteria, and then you have to go find the successful clone. And sometimes this is harder than it sounds, because the plasmid itself can go into the bacteria and grow. And so you have to be able to differentiate between the initial plasmid, shown up there on the top left, or the on the bottom left, or in the center, the successful cloned gene. So the beta globin gene is shown here in orange. We want to get it into that plasmid and grow it up and maybe do some sequencing or mutagenesis, and so we have to be able to tell the difference between the plasmid that has the beta globin and the plasmid that doesn't, shown on the bottom right. Both of these will grow in the bacteria. So how do we do it? Well, we use a test that's called alpha complementation. In this case, we have redundant tests because we have the ampicillin resistance and we also have the beta-galactosidase gene. On this plasmid in green is the gene that makes the antibiotic resistant protein. And then in yellow, which is shown here being disrupted by the beta globin gene, is the gene for beta-galactosidase. So if we have successfully cloned the beta globin gene, it is right in the middle of the beta-galactosidase gene, and it actually destroys the gene. So if you are successful, you will not have a bacteria that can make beta-galactosidase. So that's one thing that we're going to use as our screening method. So here we are with our plasmid with the inserted beta globin gene. We transform it into E. coli or some bacteria, and we plate it on a petri dish that has our selectable markers. One of them is the antibiotic ampicillin, and then the other one, well, there's two actually. There's a sugar called IPTG, which is going to initiate expression of beta-galactosidase, and then there's also a substrate called XGAL, xgal can be digested by beta-galactosidase, and when it's digested, it makes a blue color. So we're going to plate our bacteria, let them grow overnight, and then we're going to look at the color of the bacterial colonies. Some of them will be blue, and some of them will be white. If the colony is blue, that means it's making beta-galactosidase. And that means we were not successful in cloning into that plasmid. If it makes a white colony, then that means we were successful and the beta globin gene has disrupted the beta galactosidase gene. The fact that bacteria grow at all means that they have a plasmid because they are able to grow on a petri dish that has ampicillin. If they didn't have the plasmid at all, they wouldn't be able to grow because the ampicillin antibiotic would kill the bacteria. That's why we need two selection markers. So this particular slide, the next two actually repeat what we've just been talking about here. So let me skip ahead. You can read this. The bacterial colonies have to have the vector and the ampicillin resistance gene, otherwise they wouldn't grow. However, you can get the plasmid with no insert, which is not what we want, or you can get the plasmid with the insert, which is what we were trying to make. So to be able to tell between those two, that's why we have the beta-galactosidase gene. Here are the two chemicals that I told you are incorporated into the petri dish along with the ampicillin. One of them is called IPTG. This is a sugar, an analog of lactose that can activate expression of the LAC-Z gene. And then the other one is called xgal, and there's the chemical name for it there. It's also a sugar analog, but when it is coupled to a dye, and then when it's hydrolyzed, it turns into a blue color. Therefore, this is how we tell whether or not we've successfully cloned the beta globin gene. The colonies will either be blue or white, as I mentioned. Let me show you what this looks like in real life. If we look on the left, let's say we've done the experiment. There's our petri dish, and all we see are white colonies. You can see there's one colony right there that I've just circled. There's a bunch of them, but they're all white, and that's because we did not do the LAC-Z expression of beta-galactosidase. In this case, we didn't have the secondary selection. So even though we've successfully put a plasmid into these bacteria, we don't know if they have beta globin or they don't. The only way we could find out would be to grow them up and sequence the DNA, which is a lot of work. And that's why on the right you can see here, just circle this, within that circle there's a whole bunch of little blue colonies and there's some white colonies, and roughly maybe it's 50% of each. So that means 50% of our clones worked. Those are the white ones because we've disrupted the beta-galactosidase gene. But the blue ones have the plasmid with no globin cloned. So now, in this case, we want the white colonies. We would pick those, grow them up, and that would be our successful clone. So the result of cloning a gene, after this, then you can do lots of different things. One is to make a whole bunch of DNA so you can sequence it or mutate it. As the host grows and replicates its own DNA, it also replicates the plasmid, and so that's why it generates tons of more DNA. The bacteria grow it up. Just briefly to mention, recombinant DNA technology, we've been talking about cloning genes, but it can be used to clone any segment of DNA, telomeres, centromeres, repetitive sequences. Whatever you're studying, you can clone it. The very first cloning experiment was accomplished by Stanley Cohen, Annie Chang, Herbert Boyer, and Robert Helling in 1973. At this time, they already knew several things. They knew that there were some restriction enzymes that could cut DNA, and when they did, it made a piece of sticky ends that were a single strand of DNA that were overhanging. They also knew about DNA ligase, and that DNA ligase was used during DNA replication to seal DNA together. So they then figured that they could do this in the lab and make different types of different recombinant, so recombined DNA from different sources together. The very first experiment, they took two commonly grown plasmids that had been found. One's called PSC101, the other is called PSC102. PSC101 has a gene so that it confers resistance to the antibiotic tetracycline, and then PSC102 confers resistance to a different antibiotic called kanamycin. Both of them have EcoR1 restriction sites. PSC has one, whereas PSC102 has three. Here's their experiment. They grew up the different plasmids, they purified them, then they cut them with EcoR1 to generate for PSC101 either one piece of DNA, they just opened up the circle, or for PSC102 when they cut it they got three pieces of different sizes. Then they mixed them all together, added DNA ligase, and then put them back into bacteria. Those bacteria, they grew up, then they purified that DNA too, and then they analyzed what they saw. So by the different sizes of the different pieces of DNA, they could tell which plasmid they were working with and what the bacteria had. And this is what they found. So here's their first gel. Lane 1 shows you what it looks like if you take the plasmid PSC101 and you cut it with EcoR1. It linearized the plasmid, so there's only one piece of DNA, and it runs at a particular size. Lane 2 shows you what it looks like if you digest PSC102 with EcoR1. It makes three pieces of DNA, one really big one in the middle, and one's really small piece. If you take those two pieces of DNA, mix them together, and then cut them with EcoR1, you'd get four pieces of DNA of specific sizes. So if you look in lane 3, you can tell where all those pieces of DNA came from, right? You can pick out the PSC101 plasmid from PSC102. Now, when they did their experiment, mixed them together and then ligated them, put those into bacteria and grew them up, they purified another plasmid, which was their recombinant plasmid. They called it PSC105. Notice what it looks like when you cut it with EcoR1. There's two pieces. That's because they fused the small piece of kanamycin of PSC102 with the larger PSC101, and that's what this cartoon shows you here. So the fragment from PSC102 had the gene for kanamycin resistance, and that's shown in orange at the top, and then in blue is the PSC101 plasmid that had the gene for tetracycline resistance. So rather than even running it on the gel, they could have looked for this by growing the bacteria on a petri dish that had both kanamycin and tetracycline. And so this one could grow there, whereas none of the others could. So this was an exciting first cloning experiment. So we're going to now move into, for the next several weeks, our own cloning experiments. And let's talk a little bit about what we're going to some background that we need to know. So to clone DNA, you have to start with, to clone cDNA, you have to start with a sample of RNA purified from your tissue. And then in vitro, you can reverse transcribe this to cDNA. So cDNA is what DNA that's made from RNA is called, complementary DNA. Here's a cartoon showing this principle here. So on the top in red is the RNA that we've purified from our cell. You have to prime DNA for DNA synthesis. So in this case you use what's called an oligo DT primer. It binds to the poly A tail of your RNA. You add reverse transcriptase, the nucleotides, a proper buffer and the proper temperature, and reverse transcriptase will copy your RNA back into DNA. To get rid of the RNA, you add an enzyme called RNase H, which cuts up RNA but not DNA. It'll chew up the RNA, but it leaves behind a couple little strands of RNA that then become primers for the second strand synthesis, where is you add DNA polymerase and the oligonucleotides that you need, and it will make double-stranded cDNA, It'll copy it and replace the RNA primers with DNA. Now, then you have double-stranded cDNA that you could clone into a vector. In many biology labs, and most of the time you're working with cDNA, but if you're interested in what a particular protein is doing, because the cDNA represents the messenger RNA sequence, it represents the part of the gene that's called the open reading frame. You don't have to, by doing the cDNA, you don't have to worry about introns, which confuse the issue sometimes, as we saw when we looked at the genomic databases. Sometimes the piece of DNA that includes the introns can be huge. That's much harder to work with than a smaller cDNA. And also, if you have the cDNA, it allows you to make a fusion protein that you can express in a variety of different cells. Now, we talked a lot so far about restriction enzymes and how they're used, but usually now, rather than just looking for specific restriction sites and cutting your DNA up with just restriction enzymes, you can target a specific stretch of DNA using the polymerase chain reaction. This was developed in 1985 by a scientist named Kary Mullis, for which he won the Nobel Prize in 1993. You can use PCR to copy DNA without the use of restriction enzymes. You don't even need vectors or cells. You can amplify the DNA in a machine called a thermocycler. Typically, though, people take their PCR amplified DNA and clone it. PCR is ultimately relatively simple, and it's based upon the exact natural way for replicating DNA in a cell. You need template DNA, which is the region that you're interested in that you want to copy. You need oligonucleotide primers. So this is short stretches of synthesized DNA that are complementary to the region that you want to copy. I'll show you a picture in just a second. You need deoxynucleoside triphosphate, so DNTPs. This is DATP, DCTP, etc. These, of course, are the building blocks of DNA. And then you need an enzyme to copy the DNA. The two most commonly used are TAC, which was the first one that was discovered and found for this purpose, and then the other one is called PFU. Both of these enzymes are used because they can withstand very high temperatures, and so during PCR you have to heat the DNA hot enough so that it denatures and comes apart. Regular DNA polymerase, like from a human cell, cannot survive those high temperatures, but the TAC polymerase and PFU polymerase can. Here's a cartoon showing the PCR reaction. On the top left is the region that we're going to amplify, and on the right, that arrow has zoomed into an area showing you the primer site. And so the primer is shown in green, and it is complementary to the target, to one side of the target DNA. Now, if you notice, there are two primer sites, one here and one over here, because you're going to copy the DNA in the middle. This one's going to go that way. That's pretty rough looking. And then this one's going to go that way. And so it will copy double-stranded DNA in that manner. And we move down to the first step. So here's this first primer, 1B, and the DNA is being copied that way. And this is the other one, and vice versa. So now it's copied it. Notice that it goes past this first primer here. So it just keeps going, but eventually, as we cycle, it's going to start making pieces of DNA that are only from here to there. And that will be our amplified fragment. You'll see that in the next picture. So there, the overwhelming majority of the amplified product after multiple cycles will be the small piece shown in 2A1. Usually you do 20 to 30 replications during the PCR reaction, and if you do that from you will increase the amount of DNA 2 to the 20 or 2 to the 30, so up to 1 billion times from each initial template strand. So that's a huge amplification. PCR is not only used to clone, it's used to detect and quantitate the amount of RNA in living cells by reverse transcriptase. In this case, RNA is isolated from your sample, you mix it with reverse transcriptase, and you generate a single-stranded cDNA molecule that then you can go directly into PCR with and amplify. RT-PCR is super sensitive. It can detect very tiny amounts of RNA from the initial sample. Another use of PCR would be then to generate a DNA library. Um, doesn't actually have to be by PCR. You can do this by restriction mapping or restriction digestion also. Particularly if you're using genomic DNA, you would purify the DNA, cut it into a bunch of little pieces, and then clone it into a vector. A cDNA library starts with the RNA and reverse transcriptase to make a cDNA, and then that's amplified and put into the inserts. But what these do is they generate what's called a library, and each plasmid has a different piece of DNA in it. So here's an example showing you the process of making a chromosomal DNA library. So on the top left is the vector. It has a unique restriction site, probably something like ECOR1 or BAMH1. And then on the right is our chromosomal DNA with a lot of restriction sites. You would cut it up with the same restriction enzyme. So in this case, if we used ECOR1, you would cut both DNAs with ECOR1. That would generate a whole bunch of fragments from the chromosome, which you then mix with the plasmid. And then each vector would get a different piece of DNA that if everything worked perfectly, hopefully it does, you would get a representative of the entire chromosome, but in little tiny pieces. And that then is your library. You take all that DNA, you transform it into bacteria, and you grow it up. And now you have bacteria that are containing the plasmids that have your entire chromosome, but in little pieces. So that is a library. Okay, that's it for our introduction to this. I want you to do this homework just to review some of the basic concepts, and then we're going to move on to cloning in the next lecture.